Welcome to lecture number 1 on you and your hormones. This will be the first of 3 lectures dealing with general endocrinology, human hormones, and of course ending the course with applied endocrinology, reproduction. This first lecture is on just kind of general endocrinology. What are hormones? How do they function? And how are they regulated? So the system of hormones is known as the endocrine system. Endocrine glands are been categorized as ductless, which means they just secrete their product, the hormone, into the blood, not into a duct. The hormones then are circulated throughout the body in the blood, and they affect what are called target cells. And the target cells are the ones that have the receptors, the specific receptors that the hormones will bind to. As you'll see, especially in the second lecture, hormones have a wide range of functions, from dealing with increasing or decreasing metabolism. Salt balance, we've already talked about that with something like aldosterone. Growth, as in like growth hormone or the thyroid hormones, and of course reproduction, the last lecture. Now, one of the effects of hormones is that their effect is long-lasting. In other words, the effect of hormones can last your entire life. And we can compare that to the function of the nervous system, which is really fast but doesn't last very long. Hormones, of course, because it's being circulated by the blood, take their time to get there, but their effects are much longer. So again, both the nervous system and the endocrine system are communication systems. Both are being released by one cell to alter the behavior of a second cell. Both hormones and neurotransmitters require receptors and specific receptors, and binding to that receptor is going to cause our old friend, that G protein linked receptor, to alter its physical appearance and activate a signal transduction cascade. In fact, there are some neurohormones that can be used as neurotransmitters or as hormones themselves, examples being dopamine and norepinephrine, and we'll talk about those two hormones when we discuss the adrenal medulla in the next lecture. Just to hammer home the point again, the target cell must have a specific receptor for that specific hormone. If it lacks the receptor, the hormone will not be responsive, and the response, of course, could be turning off or turning on the cell. There are a bunch of endocrine glands. We'll be discussing those endocrine glands both in lecture 2 and lecture 3 mostly. Here's a nice list of many of the glands and many of the hormones we'll be dealing with over the next 3 lectures, some of which we've already talked about. If you go down to the small intestine, there's CCK, cholecystokinin, stomach, gastrin, the adrenal cortex, We're going to talk, there's aldosterone, also known as a mineral corticoid, because it activates the minerals, sodium. All right, let's get down to basics of basic endocrinology. There are basically two types of hormone. Your book has 4. I've left it at 4, but really there's 2 types. The first one are the amino acid, peptide, or protein types. Okay? Again, first classification are these water-soluble hormones, for example, the amino acid-derived ones. So we can talk about the catecholamines, peptides and proteins. A small peptide like antidiuretic hormone, larger peptide, almost a small protein like insulin, and a large protein like HGH, human growth hormone. Your book also has subdivided the proteins into a glycoprotein. This is a sugar piece on top of it. And we have a couple of anterior pituitary hormones that have carbohydrate pieces like LH and FSH. The second major category for hormones are lipids and fats. Primarily, we know these as steroids, coming from cholesterol. And there are a couple of hormones that we will talk about a little bit today and in this lecture, and then definitely with reproduction. Those are the prostaglandins. And steroid examples, we've already talked about one of them, aldosterone. And there's a couple of other ones that we'll talk about over the next through lectures. Speaking of steroid hormones, here is a biochemical pathway which produces these steroid hormones. You'll see that there's only 2 sites in your body that will produce steroid hormones. For both males and females, the adrenal cortex. And there are going to be 2 steroid produced by the adrenal cortex. We've already talked about one of them, aldosterone. And the second one is the stress hormone known as cortisol. We'll discuss that in lecture 2. The other organ is either testis or ovaries, and that, of course, enables the individual to produce the sex hormones, testosterone or estrogen, one of which is estradiol. Okay, so again, to sum up, you have polar hormones, amino acids, peptides, proteins. These are polar, they're water soluble, so they easily can be transmitted in the blood. What they can't do is they cannot cross a non-polar barrier. Oh, like a cell membrane. So, in order for these hormones to activate a target cell, they must bind to receptors on plasma membranes only. Let's contrast that with the non-polar hormones. Right, oil and water don't mix, so they're insoluble in water. Therefore, in order to move in a water-soluble environment like blood or cytoplasm, these hormones require some sort of transporter, or your book will call it a carrier. These are proteins that enable the steroid hormone to circulate in your body. However, non-polar molecules can easily move through non-polar plasma membranes. Thus, steroid hormones can get into every cell in your body. That's right, gentlemen. Right now, testosterone is moving into every cell in your body. But that doesn't make testosterone effective in every cell in your body, because what does testosterone need to affect cell performance? Correct, a receptor. Let me tell you about one exception to the rule, and that is the thyroid hormones T3 and T4. These hormones are produced from the amino acid tyrosine, but they are non-polar. Therefore, even though they're amino acids, they will work like a non-polar hormone, move into the cell directly through the membrane, bind to cytoplasmic receptors, and cause a change in cell function. It's time to go biochemistry. Let's see how the non-polar and the polar hormones work to alter cell behavior, cell physiology, cell genetics, cell everything. Ready? All right. Again, just to reiterate the point, in order for a hormone to activate a target cell, that target cell must have the specific receptor that will bind to that specific hormone. In nonpolar or lipophilic hormones, the receptors are found within the cytoplasm itself. In polar hormones, the receptors are only on the plasma membrane, because these hormones will never enter the cell. Okay, let's start with it. Again, these are nonpolar, lipophilic, hydrophobic, whatever adjective you want to use, hormones. So like the steroid hormones, so aldosterone and estrogens and testosterone and a couple others we'll be talking about. And remember those thyroid hormones that are produced from the amino acid tyrosine? They're called T3 and T4, and we'll learn about those on lecture number 2. Okay? So, the steroid hormone released by the gonads, released by the adrenal cortex, move into the blood, right? Remember ductless glands? They do not do well in the blood. In order to move, they require some sort of transport protein, a transporter, which will make them more water soluble, which allows them to circulate within the blood. Theoretically, steroid hormones will actually move into every cell in your body. You can't keep them out. They diffuse easily across the plasma membrane because they are soluble within a plasma membrane, right? Oil versus oil. So, steroid hormones get into the cytoplasm of all cells. However, in order for that steroid hormone to function, to act upon that cell, there must be a cytoplasmic carrier. Oh, sorry, got to use endocrine talk. A cytoplasmic receptor within the cytoplasm, and it will bind to the specific steroid hormone. After that, the steroid hormone binds to its receptor. That complex then translocates into the nucleus. Within the nucleus, this complex literally binds to DNA. And it binds to the DNA very close to that portion of the gene known as a promoter that promotes transcription. Bottom line, steroid hormones, with help of their cytoplasmic receptors, will regulate, which means turn on or turn off, transcription of specific genes. Okay, again, steroid hormones binding to the receptors regulate transcription directly. Okay. This slide literally explains what I just said in the previous slide. The steroid hormone, with the help of its receptor, forms a complex. This complex that was formed within the cytoplasm moves into, translocates, migrates, whatever verb you want to use, into the nucleus. There it binds to specific sites near the promoter region of the gene, where it will turn on, stimulate, turn off, inhibit, block transcription of specific genes. This is just showing you what's going on. You see the transport protein in the blood moving H, the hormone. Once it gets near the cell, it moves in, binds to a receptor, translocates inside the nucleus where it binds to specific regions of the DNA. So, steroid hormones play a huge role in turning on or turning off protein synthesis within the target cell. Examples? We've already talked about one of them. Remember aldosterone? Oh yeah, that's on exam 3 or 4, isn't it? Aldosterone works the distal convoluted tubule and the collecting duct to move into those cells within those regions of the nephron, bind to the DNA, and turn on, activate the protein synthesis of sodium pumps. Let me give you another example. Have you heard of this drug, RU486? Right? Remember, this is a very controversial drug. This drug is known as, yes, the abortion pill, produced by a French pharmaceutical company over 40 years ago. It has been a very controversial drug in the United States since the last 40 years. Given the fact that this is a class in biology and not in political science, sociology, etc., we will leave the politics of RU46 to another class. RU486 works to be a competitive inhibitor on the cytoplasmic receptor of the steroid hormone known as progesterone. We won't discuss progesterone anymore until we get to the last lecture of the course, reproduction, because progesterone is the pregnancy hormone. Okay. Progesterone makes sure that the woman is ready for pregnancy. It carries on all the things that must occur during pregnancy, including the inhibition of smooth muscle contractions of the uterus, labor. And it does that by blocking the activation of an enzyme known as prostaglandin synthase that you can see at the bottom right. Prostaglandin synthase takes phospholipids eventually and converts them into prostaglandins, and prostaglandins do all sorts of fun things. They are considered kind of a hormone, one of which is that they activate smooth muscle contractions, especially in the uterus. Another way that prostaglandins work is pain. Prostaglandins seem to activate nerve-free endings to promote pain. In fact, one of the most common drugs on the market prevents the actions of prostaglandin synthase. It is known as aspirin. Okay. The bottom left shows you what progesterone does. You see PG in the red triangles? It comes in and binds to PR, which is the progesterone cytoplasmic receptor. Together, those two, that complex moves from the cytoplasm into the nucleus, where it binds to the yellow stripe, which is DNA. Together, that blocks the activation of transcription. Again, one of the roles of progesterone is to block the production of prostaglandin synthase. Okay, so how does RU46 works? It's easy. It is a competitive inhibitor of the cytoplasmic receptor for progesterone. It binds to the progesterone receptor, PR over there, preventing progesterone from binding to it. If progesterone doesn't bind to it, that complex will not cross into the nucleus. It will not bind to DNA. It will not block prostaglandin synthase transcription. So, without the block, prostaglandin synthase will occur. Prostaglandins, the enzyme will be made. It will make prostaglandins, especially within the uterine smooth muscle. Uterine smooth muscle now can contract, and the result is the A word. So, we just talked about how steroid hormones can work directly at the level of the DNA to turn on or turn off transcription. Polar hormones, water-soluble hormones, like your peptides and your proteins and your amino acids, remember they can't cross the plasma membrane, so therefore they must bind to specific receptors on the target cells. The binding of hormone to receptor activates our best buddy, the G protein coupled receptors, activating signal transduction. I'm going to go really quick through second messenger systems. We've already talked about all three in what was it, chapter 6, I think, exam 1, I think. We'll go through it again really quick. The most common one, of course, is the cyclic AMP transduction system. And you can go through the flow chart here. We've talked about this, what happens. Examples are most of the anterior pituitary hormones work this way. The ADH works this way. So HGH, ACTH, these are anterior pituitary hormones. Don't panic. We'll discuss these in this next lecture. Again, we've seen this graph before, haven't we? Or this figure. Cartoon. Another system that is used, we discussed it really briefly. This is used by a very few hormones, like TRH, which I'll be showing you in just a few slides from now. Basically, this uses instead of cyclic AMP as a second messenger, it's using our best friend calcium. Again, activating things to liberate calcium from endoplasmic reticulum storage. The third signal transduction system we discussed briefly way back in exam 1 is the tyrosine kinase system. This is the one where when a ligand binds to the receptor, the example that the book gives you is insulin. This causes the cytoplasmic portion of the receptor to autophosphorylate as a phosphate group to an amino acid within the receptor. The phosphorylated receptor then can provide that phosphate group on to another molecule, which triggers the tyrosine kinase signal transduction system. The reason why it's called tyrosine kinase is because that amino acid within the receptor, within the cytoplasm, is always a tyrosine molecule. All right, so we've talked about hormones. We've talked about what hormones do. Now let's discuss how hormone release can be regulated. Two types. One is negative feedback, and that's the one we're going to be, and the second one's going to be positive feedback. Let's talk about negative feedback first. Okay, negative feedback, most very common, incredibly common feedback system, feedback regulation. And as the name implies, negative feedback means something is going to inhibit something else. In this case, the final product of an activated hormone can go back to and target the cells that produced the hormone. Okay? In other words, the hormone and the levels of the hormone can inhibit itself. Okay? So, this inhibition can happen in a wide range of places. Primarily, it will respond to those cells that are producing the hormone or hormones that will ultimately make the product. Let's see a couple of examples, shall we? This works so much better when I had a white board, but let me try to talk you through it. Okay, let's start with the product. Down at the bottom in the brownish box, you'll see thyroid. And the thyroid produces a couple of hormones that we'll discuss in the next lecture. One of which is a hormone known as thyroxine or tetraiodothyronine, or commonly referred to as T4. We'll explain why it's called T4 in the next lecture. Okay, thyroxine is kind of a growth hormone in a child, focusing mostly on more organs than the bones. That's growth hormone. Thyroxine in adults, after you stop growing, works with increasing metabolism. Okay, but we'll discuss how that works again in the hormones lecture next. Right now, thyroxine we're going to talk about in part of negative feedback, and thyroxine has two visible roles in that. One, it goes to the hypothalamus, where it blocks the production and release of a small peptide known as TRH or TSH releasing hormone. Thyroxine also has the ability to go into the anterior pituitary, where it again blocks the production and release of TSH or thyroid stimulating hormone. Thyroid stimulating hormone is a good sized protein, which is when released in the blood, goes to target cells within the thyroid, binds to receptors, and causes those thyroid cells to make, yes, T4, thyroxine. So again, the function of thyroxine is to regulate the production of itself by regulating the production of two hormones that will eventually cause its release from the thyroid. So why do we need negative feedback? Correct. To prevent overproduction of T4. Let's see another example. Okay, look at the bottom box. The bluish one, these are the gonads, so your testes and your ovaries. Okay. They are going to produce steroid hormones. They are going to produce estrogen and testosterone into the blood, carried by transporter proteins throughout the body. Here, testosterone will have positive effects on males and females and males, testosterone and estrogen. And we'll go into that and their effects, not next lecture, but the second lecture from now, when we talk about reproduction. These steroid hormones can get into the hypothalamus, the green box up above, and directly block production of a peptide known as GnRH or gonadotropin releasing hormone. The function of GnRH is to activate the anterior pituitary to produce the gonadotropins, which is LH and FSH. Sure enough, both estrogen and testosterone can get into the anterior pituitary, can directly prevent the transcription of the smallish proteins LH and FSH. The function of LH and FSH are they are going to be troping your gonads or activating those cells to produce the steroid hormones. Again, the function of negative feedback by testosterone. And estrogen, with an exception, and you'll hear about that in the next set of slides, is to prevent overproduction of estrogen and testosterone. So, got it? Negative feedback to prevent overproduction of hormones. Well, if we have negative feedback, should we have positive feedback? Which means the presence of the product allows more hormone to be produced. Seems intuitively wrong, doesn't it? Well, there are count them, 4 examples of positive feedback in your body. We just talked about 2 of them. Remember digestion? Do you remember gastrin and CCK? Cholecystokinin? Remember that? Those 2 hormones work by positive feedback. As long as the stomach is stretched, indicating there's food in the stomach, gastrin will be released. Positive feedback. As long as there is a meal coming into the duodenum and the duodenum is stretched, the duodenum releases CCK to allow more digestive functions within the small intestine. Positive feedback. The third one's a little strange. We'll discuss it more in the reproduction lecture, and that is estrogen. Estrogen before ovulation. So keep that in mind. However, if you look under hormone positive feedback, you will find the poster child of positive feedback regulation, known as oxytocin. Oxytocin is produced by the posterior pituitary. There's one other hormone produced by the posterior pituitary? Care to guess? Should I hum the Jeopardy theme song? You are correct. Antidiuretic hormone. So you already learned the two hormones produced by the posterior pituitary, ADH and oxytocin. Okay? Let's talk about one effect of oxytocin in females. Okay? It's 9 months. Junior wants to get out. He's tired of being in a cramped situation. He starts beating his head against the bottom of the uterus. That is known as the cervix. Within the cervix, there are stretch receptors, proprioceptors, that trigger the hypothalamus through the posterior pituitary to begin to release this small peptide known as oxytocin. Oxytocin moves through the blood, binds to oxytocin receptors on smooth muscle on the smooth muscles surrounding the uterus, causing smooth muscle contractions, which we call, labor. Right? Those weak, smooth muscle contractions? Right, weak, smooth muscle contractions. Okay. As long as Junior is beating his head against the cervix, the positive feedback loop will occur and oxytocin will be continually released by the posterior pituitary to stimulate smooth muscles within the uterus to contract, forcing Junior out. As soon as Junior leaves the uterus and got to breathe on his own, oxytocin release will finally stop. Positive feedback. Positive feedback continues. Junior's hungry. Junior starts nibbling on mom's nipples. Mom's nipples also have proprioceptors that also trigger the hypothalamus and the posterior pituitary to release oxytocin. Oxytocin's effects here are, again, smooth muscle, to squeeze the mammary glands where the milk is, and milk will squirt out of the mammary glands through the nipple into Junior's mouth. As long as Junior is suckling, as long as Junior is stimulating the posterior pituitary to release oxytocin, positive feedback will continue. One note, oxytocin functions to release milk, not to produce milk. The milk production in mammals is caused by another hormone, this one from the anterior pituitary, known as prolactin. We'll talk about prolactin in our next lecture. Oxytocin. Okay, so we talked about the classic cases of oxytocin, positive feedback in labor and in milk release. So, a couple of questions. Do males have oxytocin? Yes, males have oxytocin, and oxytocin in the male is released by the posterior pituitary. Okay. There is no labor, there is no milk production in males. So what does it do? Unknown. Except for one interesting study. Anyone ever own a rabbit as a pet? Rabbits? What happens when you put a male rabbit with a female rabbit? I give it about 5 seconds. Right? Male rabbit sees female rabbit, smells female rabbit, male rabbit. There's a reason why there's a lot of rabbits in the world, shall we say? Study done, 20 years ago, where they blocked oxytocin receptors in the rabbit, put male rabbit with the female rabbit. The male rabbit ignored the female rabbit. So, possible function number one in males, and seemingly also in females, is sexual arousal. Oxytocin has been studied by lots and lots of behavioral ecologists and lots of psychologists. And it shows that there is some sort of, I hate to use the word, love among its effects. Let me give you a couple of examples. Number one, it has been shown using oxytocin nasal spray in college students, both males and females, that higher levels of oxytocin increase visual attractiveness of one sex looking at the other, or same sex, I guess. Okay, is that love? Attraction? They have examined the bonding effects in a rodent known as a prairie vole. Prairie voles are unique, especially among rodents. They pair bond for life. Once a male and female meet, get together, they do not leave one another unless one participant gets killed. It has been shown that the levels of oxytocin increases dramatically when the pair are together. Love? Another study, taking blood samples out when a pet owner, a dog owner, begins to pet their dog. The petting has to be between 5 and 20 minutes. They take a blood sample, and oxytocin levels have risen dramatically after the petting session is over. Interestingly, both in the human owner and their dog. So, bonding, love. There's been a new field that has opened up called neuroeconomics, where sociologists, psychologists, business associations have been measuring the effects of oxytocin in trust and generosity. What they do is they usually play some sort of trust game, or if you're very generous, usually this is college students, and usually this is with or without a nasal spray containing oxytocin. And then they measure, and it is turned out that oxytocin levels, when increased using this nasal spray, contributes to an increase in both generosity and in trust. Love? Question mark?